Welcome to another deep dive. Today we're going to be looking at organoids. They're these 3D structures that mimic organs and are changing how we study human biology.
Mini organs grown in a lab. That's pretty amazing.
Right. We'll be focusing on an article from Assay Genie, a company that makes tools for life science research.
Okay.
The article is called Optimizing Organoid Culture Conditions, Paving the Way for Revolutionary Advances in Biomedical Research. It was published February 14th, 2024, and was written by Tafreem Ali.
I see.
She has a background in bioinformatics.
Cool. So this Assay Genie article,
Yeah.
it goes really in-depth about making these mini organs, the challenges and the possibilities.
Yeah, they really highlight how important it is to fine-tune the conditions for growing them.
Okay.
Like baking a cake.
Yeah.
You need the right ingredients and environment.
Makes sense. So let's start with the ingredients, stem cells, but which types are we talking about?
Okay, good question. So you can choose pluripotent stem cells.
Hmm.
They're like wild cards. They can become any type of cell in the body. Or you can use adult stem cells.
Okay.
They're already on a specific path, like becoming a liver or heart cell.
So the choice of stem cell depends on what you're trying to do with the organoid.
Exactly. Like if you're studying how a heart develops, you might pick a pluripotent stem cell.
And guided to become heart cells.
Yeah, and then imagine you've planted your stem cells.
Okay.
They need the right soil to grow.
Okay.
That's where the extracellular matrix comes in, or ECM. It's like scaffolding for your organoid, providing support and signals that tell the cells how to behave.
So we're not just throwing cells in a dish and hoping for the best.
No, definitely not.
Yeah.
So what kind of materials make up this ECM?
Well, scientists often use things like Matrigel, which comes from mice,
Okay.
or collagen, to mimic the natural environment of tissues.
Interesting.
But there's also excitement about synthetic ECMs.
Oh, why that?
They offer more control over the environment, like a custom-made support system.
That's pretty amazing.
Yeah, it's like we're architects of the microscopic world.
But cells need more than just support, right? They need instructions to become specialized.
Yeah, exactly. That's where growth factors and signaling molecules come in.
Okay.
They're like messengers telling cells, hey, you become a heart muscle cell, you become a blood vessel cell.
Who they orchestrate the development of the organoid.
Exactly.
This sounds incredibly complex. What are some challenges in getting this signaling right?
Well, getting the concentration of growth factors correct is critical.
I can imagine.
Too much or too little of a certain signal can throw everything off.
Okay.
Think of it like a recipe.
Right.
A small change can really change the final dish.
So we've got the stem cells, the ECM, and the signaling molecules.
Right.
But what about basic needs, like oxygen and nutrients? As the organoid grows, I imagine it's difficult to get those to every cell.
That's a huge challenge.
Yeah.
In traditional cultures, the cells on the outside get the resources, and the ones in the core get starved.
Oh, so it limits the size.
Yeah, and the complexity.
So how do scientists deal with that? How do you make sure every cell gets what it needs?
One approach is to create dynamic culture systems.
Okay.
Think of them like high-tech nurseries for organoids.
Interesting.
For example, spinning bioreactors use constant motion to ensure even distribution of nutrients and oxygen.
So it's like creating artificial gravity.
Sort of.
Wow. So it's not just creating these mini organs,
Right.
but creating a whole environment to support them.
Absolutely. And another exciting approach is using microfluidic devices.
Okay.
These are tiny channels. They mimic the flow of fluids in the body, delivering oxygen and nutrients.
That sounds like a big deal.
It is.
Yeah.
We're mimicking the body's processes to grow these complex structures.
But even with this, I imagine there are still some hurdles.
Oh, yeah, definitely. It's not all smooth sailing. These dynamic culture systems are really something else.
It's like we're engineering these tiny worlds to help them thrive.
Yeah.
But I imagine even with all this, it's still tough to replicate the full complexity of an organ.
You're right. One of the biggest challenges is recreating the cellular diversity.
Okay.
Think about your liver.
Yeah.
It's not just a bunch of the same cells. It's got a complex architecture
Yeah.
with all these specialized cell types working together.
So how do you even approach that? Wait. Is it about guiding different stem cells to become all those different types?
Exactly. Researchers are figuring out how to precisely control the differentiation, guiding them to create all the different types needed.
He's like conducting an orchestra.
Yeah.
Making sure each cell plays its part.
A microscopic orchestra.
And I remember from the Assay Genie article,
Yeah.
challenges with creating vascular networks.
Oh.
It's like building a city with no roads.
Oh, yeah, that's a good one.
Things get congested fast.
As organoids grow, They need a way to transport oxygen and nutrients, just like a real organ.
Right to the core.
Without those networks, the center cells starve.
So the organoid can't grow as much.
Exactly.
So what's the solution? How do you get blood vessels in there?
There's so much research happening.
Okay.
Scientists are experimenting with growth factors and signals to stimulate blood vessel-like structures. Some are even using bioprinting to guide the development.
It's like combining all these cutting-edge techniques.
Yeah, it really is remote.
And make these intricate structures.
And this opens up so many possibilities for medicine.
So let's talk about those possibilities.
Okay.
The article talked about personalized medicine. Can you tell me more about that?
Imagine taking a patient's own cells and creating an organoid that reflects their biology to test different treatments.
So it's personalized for each patient.
Yeah, like a personal avatar.
Helping tailor therapies.
Exactly. We could study how a new drug might affect their specific cells.
Wow.
Or predict their response to treatment.
That's incredible. It's like a crystal ball.
Yeah, helping make more informed decisions.
So we're moving away from that one-size-fits-all approach.
We are.
And I bet organoids are also great for drug discovery.
Absolutely.
Yeah.
Traditionally, new drugs were tested on animals.
Right.
But they don't always reflect human biology.
Of course.
Organoids offer a more human-relevant system.
So potentially we reduce reliance on animal models?
Yeah.
And develop drugs faster?
More efficiently, too?
And you can create organoids from different cell types, right?
Yes. So we can test how a drug affects various organs, like how it's metabolized in the liver.
It's like a miniature clinical trial.
In a dish.
Revolutionizing drug development.
It really is.
And what about regenerative medicine?
Right. Organoids can be used for repairing damaged tissues.
Okay.
Or even growing whole organs for transplantation.
That sounds like science fiction.
It's getting closer.
Are we talking about growing whole organs?
Not quite yet, but scientists are making progress. There are studies using organoids to repair tissues in animals.
Interesting.
And they're looking at ways to scale up production and create more complex structures.
So maybe someday we rely less on donor organs.
Maybe.
And more on these lab-grown ones.
It's a possibility. The implications are huge.
We could address the shortage of donor organs.
And save so many lives.
This is incredible, but with all this advancement, we have to think about the ethical implications.
Absolutely.
Right.
We need to be mindful of the risks.
Right.
And use this technology responsibly.
And what about CRISPR-Cas9?
Oh, right.
The article mentioned that
CRISPR is amazing.
it's a gene editing tool, right?
Yeah. It lets us make precise changes to DNA, like molecular scissors that cut and paste genes.
So how are they using CRISPR with organoids?
One exciting thing is creating models of genetic diseases.
Okay.
We can introduce specific mutations into the organoids.
So you're recreating the genetic defects
that
cause the diseases.
Instead of studying the disease in a patient, we study it in a dish.
I see.
In a controlled environment.
So you can really study the disease process.
Yes. How the mutations affect development and function. And even screen drugs.
To see if they can fix the defects.
Exactly.
Like fast-forwarding evolution.
Sort of.
Seeing these changes on a miniature scale.
And this is huge for developing personalized therapies.
For genetic diseases.
Yes. We could even use CRISPR to make organoids genetically matched to a patient.
Wow.
To test treatments on their cells.
This is really personalized medicine.
It is tailoring therapies to their genes.
And you could use CRISPR to enhance organoids too, right?
Scientists are using it to promote blood vessel formation.
Oh, wow.
Getting closer to functional vascularized organoids.
Adding another layer of complexity.
Yeah, another piece of the puzzle.
This has been an incredible journey.
It has.
From basic building blocks to revolutionizing medicine.
And there's still more to explore.
Right. In the final part, we'll look at how organoids are being combined with other technologies.
Like organ-on-a-chip systems and machine learning.
We'll also discuss the future of organoid research.
And the potential for understanding human biology.
And transforming medicine.
Exactly.
So join us for the final part of our deep dive.
We'll continue exploring this fascinating world.
And the impact on healthcare. Welcome back. It's amazing to think we can recreate human biology in a lab.
I know, right?
Using these mini organs.
And the field is moving so fast.
We've gone from basic cultures to systems that mimic the body.
Exactly. And today we're going to see how organoids are being combined with other technologies to push research even further.
One that I'm really interested in is organ-on-a-chip systems.
Oh, yeah, those are fascinating.
Imagine a tiny device
Hmm.
that recreates the environment of an organ
With channels that mimic blood flow.
and nutrient exchange.
Yeah, so it's not just growing the organoid alone.
Right.
It's creating a system that mimics the body.
Exactly. And you can use them to study how different organs interact.
Right. Do you have an example of how they might use this?
Sure, imagine you're studying a drug
Sure.
that's taken orally.
Uh-huh.
You want to see how it's absorbed in the gut and then processed by the liver.
Right.
You could connect a gut organoid to a liver organoid.
On the chip.
Yeah. And mimic the flow of the drug.
So you can see how it affects multiple organs.
At the same time,
Yeah.
it's a more comprehensive picture.
Absolutely.
Yeah.
This is huge for drug development
Right. You can test safety and efficacy.
in a more human-relevant system
It's like a mini clinical trial.
on a chip.
And you could use these systems to study diseases too, right?
Oh, yeah, definitely. How they progress and affect different organs.
Researchers are using them to study cancer,
Yeah.
infectious diseases,
All sorts of things.
to see how they spread.
It's groundbreaking stuff.
What about machine learning?
Oh, that's another big one.
How is that being used?
Well, researchers are using algorithms to analyze images of organoids.
Okay.
They can spot subtle changes that might indicate a disease.
So they see things we might miss
Exactly. And they can help predict how an organoid might respond to a drug.
based on its characteristics.
Right. It's like having a crystal ball.
Helping make decisions.
Exactly. Machine learning is also used to optimize those culture conditions.
Oh, right.
Personalized treatments. Even design new organoids with specific properties.
We're using AI to understand these systems.
It's incredible. It really is.
This deep dive has been amazing.
It has. We've seen how these mini organs are changing research,
Opening up possibilities for personalized medicine,
drug discovery,
even regenerative therapies.
and as the field keeps growing,
We're going to see even more incredible discoveries.
new applications. The possibilities are endless.
It's exciting to be following this field.
It is.
It has the potential to transform healthcare.
Offering new treatments and cures.
This technology is truly remarkable.
It is.
Thank you for joining us on this deep dive.
We hope you enjoyed it.
We hope you learned something new.
And until next time, keep exploring.